Hello again, and welcome to this lecture on genetics, epigenetics, and addiction, an introduction to these ideas and these terms. This is for the ARS 3325 course on the family dynamics of addiction. And I'll just preface this lecture by saying that I am not a geneticist, and we don't expect anyone taking this course to result in being a geneticist who understands all of this perfectly. We want to give you a brief introduction to important terms and concepts related to genetics and epigenetics and how those might relate to addiction and recovery. So with that, let's begin talking about genes and what they are and what they're not. Your book chapter that you read makes the statement that genes contribute to behavior, but they're not responsible for behavior. Another way of saying that is that genes influence us heavily, but they don't cause anything specific to happen in most circumstances. And so it's important for us to understand that as we look at genetic studies and as we understand genes and addiction, that our understanding will always need to be coupled with our understanding of the environment and other things that are going on, rather than just say this gene 100% caused this. And how the environment will come in and interact and play a role, we'll talk about a little bit later in this lecture as we get into epigenetics. So what is a gene? Well, genes are usually thought of as DNA, information encoded in DNA, which is deoxyribonucleic acid. Now, some of you are probably starting to freak out, thinking, I didn't get into this course to get back into biology. That's okay. We're not going to get too much into biology, but you may remember DNA, or deoxyribonucleic acid, from your biology class. Basically, it's just a gene structure that's made up of nucleotides. And those nucleotides in that gene structure stay at all times in the cell nucleus. Nucleotides, for those trying to remember their biology, are adenine, thymine, cytosine, and guanine. A, T, C, G. And they create pairs and they match and they create these strands of DNA that contain information that tells cells what to do. It's important to note that humans have only 23 pairs of large strands or long strands of DNA. However, only a portion of those strands actually code for genes. Most of the information in that strand does not. Now, what does that part do? I don't think we know fully, or I say we, the geneticists, the scientists, I don't think they fully know. In fact, some of that part of the unused strand that's not used for genetic coding is called junk DNA. Other parts they know plays a supporting role in the process of how DNA influences the functioning of a cell, but we don't know exactly. Anyway, an important process and point here is to note that DNA tells a cell what it is. It's the instruction, it's the code, it's the programming for a cell and what that cell is and what it's supposed to do. And kind of a unique fact I think is interesting is that most cells are pretty much identical until DNA tells them what type of cell they are, and through transcription and translation processes that we're going to talk about, tells that cell what it's supposed to be. This is interesting just as a family therapist coming from a systemic background, thinking about family dynamics, thinking about addiction. It's interesting, some people believe that sibling order in the family therapy field impacts how we find roles in our family and develop personalities and why we are the way we are. Similar to a cell, right? It's born, it's nearly identical to everyone else, but through the environment, the body tells this cell, or DNA tells the cell, what is needed at that point in time, and that's what it becomes. That's not a perfect correlation there and perfect metaphor, but it's interesting to think about people that way. How often do we become what we are because the environment is sending us cues to take a certain role or to perform a certain function? And the body may work in similar ways. So let's talk about these transcription and translation processes. Transcription is the production of ribonucleic acid, RNA, from a DNA strand. Ribonucleic acid, if you remember from biology, is basically just that, a copy of deoxyribonucleic acid, only without the deoxy. So RNA is produced in the process of transcription. Why is this important? Well, for whatever reason, DNA never leaves the cell nucleus, if you remember from just a minute ago when we told you that. RNA, however, can leave the cell nucleus. So if DNA is going to tell a cell what type of cell it is and what function it has, what it's supposed to be, then somehow it has to get out and share that information with other parts of the cell. RNA is the mechanism through which this occurs. And that process is the transcription process. So as mentioned, RNA can now leave the cell and carry information and translate it, this is a process called translation, to ribosomes in the cell. Now, ribosomes play an important role in the production of proteins that influence cell type and cell functioning. So at the end of the day, it's these proteins that are made by the ribosomes that make that cell what it is, that turn that cell into an eye cell versus a skin cell or a bone cell or whatever it might be. And that's all driven at the core by DNA. Perhaps the best way to remember all of this, if you're starting to freak out, is just that DNA makes RNA, which makes protein. That's in your book. I think they highlight that phrase. DNA makes RNA, which makes protein. At the end of the day, you and I are all just a big batch of protein as far as our physical entities are concerned. Let's talk about a genome as well. The genome is a complete set of genes or genetic material that's present in a cell or an organism. If you look at the graphic on the right, you'll see a bunch of A's and G's and T's and G's and C's, and they're all lined up in sequences and rows and columns. Basically, this is an example of a genome where the nucleotides from a DNA strand are kind of lined up in rows and columns in the sequence they're in. And then geneticists and scientists can look at this and they can see patterns, they can see differences between people and why, and from these differences they can learn more about both strengths and diseases that might occur. Just one example to show you how this can be a positive thing. Recently the Human Genome Project was funded by the federal government, and one of the things that's come out of this, being a therapist, this is an example that comes to my mind, is that people with major depression, for example, who are really struggling, or some other mental illness, and they've gone through this process of trying to find a medication that will help this disease, and they keep getting medications that are having adverse effects, they can get a DNA test and have their genome analyzed, and the results of that can tell them which medication should work the best, should be metabolized the best by their body, which should influence the efficacy of the medication, thus eliminating lots of years or months or whatever it might be of suffering and experimenting. But anyway, that's just one example. And let's move on and talk about polymorphisms. Polymorphisms are the presence of a genetic variation within a population. So when the sequence is different in certain areas or in certain rows or columns, this is a polymorphism, and I mean different between populations or between people. Scientists believe that these variations are what account for adaptation and how people adapt and grow, explaining partially the process of evolution. But polymorphisms can also be negative in that those differences, those genetic variations, can be signifying of a disease of some type. So anyway, we'll leave it at that. Genome, polymorphism, both cool and important stuff. But let's move on to the last part of our lecture about epigenetics, how genes and environment can interact to influence our behavior. Epigenetics is often also referred to as imprinting. If you see that written in the literature, they're probably talking about something similar to epigenetics. But it's the study of changes in organisms caused by modification of gene expression rather than alteration of the genetic code itself. So if you look at this diagram at the bottom, you got your DNA in the cell nucleus. It's transcribed into RNA, which goes outside the cell nucleus, and then the RNA, through translation processes with ribosomes, creates proteins, and that again helps inform the cell what type of cell it is and what its function is. At the very bottom of that, at the bottom of the slide, you'll see that this is the process of how a genotype turns into a phenotype. Our genotype is the information at the core of our cell, let's say this is an eye cell, then the phenotype would be what color of the eyes we actually see. What are the proteins in the eyes and what color and do they take and how do they look? Anyway, just as an example of epigenetics, this process can be altered by the environment as it happens, and phenotypes can change even if genotypes do not. For example, there are a few people, so I believe, so I've read, that their eye color will change throughout their life. Mine do not, and most people's do not, but some people's do, and this is probably partly explained by something going on during transcription and translation processes that might have an effect there. And there might be better examples that would be more applicable to most of us, but we'll leave it at that for now. So gene expression is the phenotype, how a gene expresses itself as a phenotype. So it's the process by which a gene leads to the appearance of a phenotype and of a corresponding character. And I think I'm kind of reiterating a few things here, so I hope you understand that, and I'll kind of move on. Another important aspect of epigenetics is that we talk about the epigenome. This picture is kind of hard to see, but you've got a genome there at the top, it's the DNA strand, and then you've got an epigenome there in the middle. And epigenomes are usually attached to genomes. They're proteins and other chemical compounds that can tell the genome what to do. They can tell it to stretch out so that it's easy to make a copy of RNA, or they can tell it to coil up, in which case it's more difficult to make a copy for RNA. And so they communicate with the genome, at least chemically, and they influence what information gets passed on or what information does not get passed on. One important genome is called the histone. It's a protein that the DNA is wrapped around, and it's definitely involved in helping DNA uncoil and coil up during the transcription process so that it can get all the information copied for the RNA. Now, due to environmental information and experiences, histones can be altered, and then the way they interact with the DNA gets altered. And so at the end of the day, scientists say that histones work like switches. They turn certain parts of the gene on or off, as far as it doesn't erase existing information in the DNA, but it turns certain parts on or off so that when information is transcribed and translated and resulting proteins and phenotypes come about, it might be missing information or having all the information. It depended which switches were turned on or off by the histone. The formal definition is any group of basic proteins that is found in chromatin, which is this DNA strand. Okay, so these are some brief insights and thoughts and introductions to epigenetics built on top of genetics. And I just want to leave you with one metaphor that might help explain this if it's kind of been a difficult concept. Think about genes or our genome or our DNA as the text in a textbook that's been printed at the publisher. It's there, it's permanent, it's not going anywhere. No matter how many people I hand it to, that type should be there. And I should be able to read it and get the information that the book was originally meant to give me. However, through experiences and translational processes, and I guess in this case, this book is about advanced potion making. Someone's going to school to be a potion maker. They take their textbook, and throughout their courses and their experience in interacting with peers and instructors, they make notes, they make footnotes, they write side notes, they highlight things, they underscore things, and they add information to the original text that will help them turn certain parts of the text on, so they study it for the exam and ignore other parts of the text, etc. This kind of explains epigenetics and histones that are turning on certain parts of information and turning other parts off, highlighting certain information and unhighlighting or whiteouting other parts of information. Now imagine this person who's studying potion making graduates, and they want their child to follow in their footsteps, so they hand them this book as they go to school. Well, that child not only has the original text, the original DNA, they also have all the side notes and the footnotes and the additional information that's been written in to guide them through their process. Maybe that information is good or bad. Maybe someone defined something and wrote it in the margin of their book, and that information gets passed on to the child, well, that's a disadvantage. But other information passed along could be an advantage to that child. And then so on. Let's say that child adds additional information. They cross things out or update things or revise things, and then they pass that on to their child, and so on and so on. This is the idea behind epigenetics and intergenerational transmission. When we talk about how addictions are passed along, maybe there could be a note on the side of a page of the human textbook of DNA that says, hey, if you're stressed or in pain, or if you've been through trauma, one good way to medicate this is through illicit drugs, or through drugs that make you feel really good or that release dopamine. And that information is passed along and is encoded in a way into genetics, not in the core, the DNA, but in the other parts of the process. And so basically, at the end of the day, we can pass information along, revise it, change it, and at the end of the day, none of the original text, the DNA, is altered. We're just writing on top at this point. Now, an important point to make on that is, let's say that the print is printed in ink and it's there to stay. Let's say the notes are written in pencil, and over time, handing the book, opening, shutting it, putting it in backpacks across generations, some of that writing starts to fade. That's true of epigenetics as well. While DNA is unchanged and passed from generation to generation to generation, how it is used and how it is expressed changes. And they think that epigenetic information is only passed along for about 3 to 4 generations before it's out and no longer in that textbook. Anyway, I hope that metaphor is helpful. An example of that metaphor is when 9/11 happened. They've done studies on, I think you may have seen this in your documentary, that mothers who were expectant at the time of 9/11, their children have elevated cortisol levels even though they weren't alive necessarily and conscious during the traumatic event that has influenced anxiety in their mother and now them. Other examples would include, again, passing on addiction, but more positively, passing on experiences of recovery. Maybe that paragraph about using drugs to cope with stress, pain, and trauma gets crossed out by someone in the next generation, and then there's a new note that talks about recovery, or here's how to cope with your problems or your skills, and that information would get in there and passed on to the next generation as well. Anyway, I am going on and on. I'm going to end this lecture there. Hopefully this gives you a few ideas into the basics of genetics and epigenetics and all the other stuff we've talked about, and I wish you a good day. Thank you.